So the study that I'm going to be talking about is a study that we conducted last year, in collaboration with Caitlin Sarlo at the USDA Agricultural Research Service in Ames, Iowa. So the focus of this study is working with Klebsiella. Why Klebsiella? So Klebsiella is a very important pathogen associated with mastitis. Okay, we know that mastitis is a very important disease to the dairy industry. And usually, Klebsiella is a pathogen that causes really severe and chronic cases in mastitis. Most of these cases are going to be treated with antimicrobials, and since we are focusing on reducing the usage of antimicrobials, it's important to know how we can actually know more about how Klebsiella actually works. So, why did I choose Klebsiella? Because during my graduate work, we did several randomized clinical trials where we looked at the quarter somatic cell count of these cases, comparing them to, say, E. coli, for example, and we saw that over time after these cases were enrolled in the study, SEC remained elevated for the entire time, right? So that compared to E. coli or culture negative. Then when we look at whether the Klebsiellas were the same, we isolated them over time, every week. And actually, if you look at this gel for cow 1 in column 1, 2, and 3, the column 1 is when we enroll the case, and that's the Klebsiella pneumoniae that we isolated. 2 and 3 were follow-ups, but what we observed pretty much was that it was the same pathogen, the same Klebsiella, which meant that when you compare Escherichia coli to Klebsiella, Klebsiella really induces a more chronic intramammary infection, so it stays there, it never leaves, right? So, and also it was associated with more cooling and more voluntary quarter dry-off, so worse clinical and treatment outcomes, which is bad for farmers because they usually never want to know or have these specific pathogens causing diseases in their cows. Now, this was the collaboration that we had with Caitlin Sarlo at Iowa. They have a really unique resource, which is a specific herd of cows that I'll call them just 1964 Holsteins. They haven't been subjected to any selection for milk production, so they're pretty unique, and they have also contemporary Holsteins which they have been selected. So the idea is to compare both groups, so the 1964 cows and the contemporary Holstein cows after they have been challenged in 1 quarter with Klebsiella pneumoniae, and compare and quantify the cytokine production. Okay, that was at least my part. Okay. This was some materials and methods. 2 experimental groups. On day 0, we challenged 1 quarter per cow in these 2 groups, and then we collected samples, IM and PM, for approximately 4 days, starting day 0, 1, 2, and 3. We had a lot of samples, and we have a lot of results, and I just want to point out that here I'm presenting some preliminary data, okay? We still have a lot of data to process and analyze for you to see, okay? And when I look at the physiological response, I wanted to look first at the rectal temperature. We also look at the somatic cell count and CFU. CFU was important because we wanted to make sure that the quarters actually got infected after the challenge, right? And when we look at whether there was an impact of the experimental group into rectal temperature, somatic cell count, or CFU, we actually did see that for rectal temperature and SEC, there was an effect of group. For the most part, for the rectal temperature, we saw that in the 1964 cows, yeah, it was greater, if I see correctly. Same thing for the somatic cell count. And for CFU, we didn't actually see any effect of the experimental group, which was good because at the end of the day, the only thing that we wanted to see was whether there was an impact of the experimental group, okay? Then when we look at the cytokine production, again, we measure maybe 15 analytes, is what's called different cytokines. And here I'm just presenting a few of them, okay? So IL-1 alpha, IL-1 beta, IL-1-4, IL-1-6, IL-1-8, and IL-36, sorry. So for some of these, if you can see, is that IL-1 alpha, beta, and IL-1-4 maybe with a tendency, contemporary groups, the least square means was greater in those groups compared to IL-6 and IL-8, where we observed that 1964 cows, the values were greater, okay? Now, IL-10, which is my favorite because it's a cytokine, but it actually regulates the immune response, which I think is very interesting because it may play a role in actually regulating this type of infections in cows. We saw, for example, that there was maybe a temporal difference in when they pick. However, when we look at just combining the results, we didn't have enough power to look into those temporal relationships, but overall, both groups were actually able to mount the immune response. Now, as I will point out later, I do want to focus on IL-10 because it may be a potential for the future for regulating the immune response because it actually regulates other cytokines, right? In future trials, what I want to do is to do challenge using Klebsiella, but taking more frequent samples because here, what is lacking is understanding more the temporal relationship. And for that to happen, we really need to do this more frequently. And I think that's it.
Thank you.